Sanger sequencing method, or chain termination sequencing method, is first-generation sequencing method. Use ddOxy nucleotides that lack hydroxyl group and incorporate with growing DNA strand during PCR reaction as DNA chain terminators to generate fragments with ends ddATP, ddGTP, ddTTP, and ddCTP, as mentioned in next slide. Continuously. This slide referred to chemical structure of dNTPs and ddNTPs. As we show, dNTPs contains hydroxyl group, while ddNTPs contains hydrogen instead of hydroxyl group. Manual Sanger sequencing method. Initiate by DNA extraction and denature DNA to obtain single DNA fragments and divide DNA sample into 4 tubes with primer and dNTPs and DNA polymerase and 1 ddOxy nucleotide in each tubes to terminate DNA synthesis at specific base to create set of DNA fragments with different length, as mentioned in next slide. After that, separate amplified fragments in parallel lanes, in denature polyacrylamide gel electrophoresis, and autoradiography gel in X-ray film, or UV light, to visualize length of amplified fragments, and red bands from bottom to top according to direction of DNA synthesis, to compare sequence with reference sequence, as mentioned in next slides. On the other hand, automatic Sanger sequencing method, fluorescently label primer at 5N to amplify specific DNA regions, and label each of ddOxy nucleotides with fluorescent dyes, and run reaction in single lane in denature polyacrylamide gel electrophoresis, to visualize nucleotides peaks that reflect sequence of chain termination, to compare output sequence with reference sequence, as mentioned in next slide. Finally, Sanger sequencing method. Use single-strand DNA for sequencing, and primer to flank regions of target sequence. And Taq DNA polymerase to synthesize new DNA strand by adding nucleotides to primer. And deoxynucleotides. Add in synthesis process of DNA strand. And ddOxy nucleotides. Used to interrupt DNA strand synthesis. As chain termination strand.